Okay, welcome everybody. I'm going to go through the sheep brain dissection that you're going to do next week. Today's the October 18th, Friday, and a week from today, October 25th, we'll be meeting in the Vet Tech building. And again, that is going to be in room V as in Victor 106. We'll meet around 11:40 or we'll get started around 11:40. We could probably get in there a few minutes early. And it's going to be a packed menu, a packed schedule. So we're going to go, I'd like to be done by 2. So if you have to leave before then, that's totally understandable. If you feel woozy or queasy or you need to step outside or for whatever reason, again, um, it's structured, but you're not on lockdown or anything. So anyway, let's take a peek at the sheep brain. Some of you have possibly dissected a sheep brain. I know I'm teaching a high school freshman biology class this semester, and several of them in middle school dissected a sheep brain. I think in 8th grade. So, and I'm like, wow, cool. I took an A&P class in high school, anatomy and physiology class in high school, and we didn't do anything like that, let alone in 8th grade. So if this is repetitive or redundant, so consider it a review. But here are the primary structures that we're going to look at. And next week on Friday, I will have not only this pulled up, but I'm going to have let me go back. I'm going to right click, open that in a new tab. All right. So the slideshow that I have is going to go with the checklist. So next week, um, if you do bring your laptop or tablet, phone, maybe. I mean, you use your phone. I don't know. Anyway, the sheep brain isn't real. There's not a lot of spritz and ooze and gore or anything. So you'll have gloves on. But if you want to have your laptops and you want to have this open as you go through it, or if you want to have the PowerPoint open. Either way, I will have these on the big screen as well. So if you bring nothing with you except yourself, then that's fine too. So just know that this will be on the big screen next week. So anyway, hopefully all of you have gone through the nervous system. I think at this point you should be heading into getting ready at least to think about heading into cardiovascular. So anyway, some of these terms should be familiar. The first 3 there are going to be the meninges or the coverings of the pretty much the entire central nervous system, so the brain and spinal cord. But we're going to see dura mater, arachnoid mater, and pia mater. Now, the dura mater is going to be dura means durable or tough. So, uh, uh, mater actually means mother. So, dura mater means tough mother. So that's going to be we're going to see the brains are going to be covered in dura mater, and that's what's going to be connected to the underside of the skull. So obviously these are no longer in the skulls, but if they were, the dura mater that we're going to remove, that's going to be the first thing we do. That's actually the basically in contact with the skull. And then so we'll remove that first of all. That'll be the most striking feature right away when you get your sheep brain. And then the arachnoid mater is the middle layer. Again, these are the meninges. And the arachnoid mater is the middle layer. Arachnoid, of course, has to do with spider or webbing. So we may see a little bit of webbing once we remove the dura mater. And then up against the brain itself, so we remove dura mater, arachnoid mater usually comes with it. Pia means soft, and so we're going to see when this is removed, the brain is going to be very shiny. There's going to be like a little sheen, kind of a clear sheen layer, almost like a varnish over the top of it. And that's the pia mater. Again, that means soft mother. So from superficial to deep, we're going to see the three meninges, and then we'll look at the inferior aspect or the underside of the brain, and we're going to see the pituitary gland, and then actually, when we take the dura mater off, oftentimes the pituitary gland will come with it. Um, so which we'll see. And then optic chiasm. Chiasm means cross. So we may see where the optic nerves form kind of an X. So chi in the Greek alphabet is kind of like a letter, looks like a letter X. It's a k sound in the Greek alphabet, but the symbol is an X. So anyway, optic chiasm is a kind of an X-shaped looking bundle of nerves that forms the optic nerves that go out to the eyeball. So again, we see it cross, so that means the right optic nerve actually goes to the left side of the brain and vice versa. And then we'll also see on the underside of the brain the olfactory bulbs, and those are for the sense of smell. So a couple of cranial nerves. Again, when you reviewed the central nervous system, actually the nervous system as a whole, you saw that the brain and spinal cord make up the central or CNS, and then all of the nerves make up the PNS or the peripheral nervous system. So these are a couple of cranial nerves that are part of the peripheral nervous system. So we'll see those as well. Um, and then we have four uh, different lobes of the cerebrum. So you'll notice these are actually named for the skull bones that they and the regions as well. So the skull bones, the regions of the skull, the regions of the cranium, and the regions of the brain all have the same terms. So frontal lobe we'll see is going to be toward the front of the cerebrum, and then the temporal lobes we'll see along the sides, and the parietal lobes we'll see on top, kind of like the crown of the brain, and then the occipital lobe will be toward the back, more or less kind of the base of the brain. As we're looking at the cerebrum, we'll also see uh, plural would be gyri. So if you play Animal Crossing, right, like the gyroids, but uh, but anyway, the gyrus and then the sulcus. A gyrus is actually kind of you've probably all seen the brain, especially it's Halloween time. You maybe definitely are seeing image illustrations of brains or maybe models of brains. Maybe you have some at your house, or you certainly see them at the store. They get little gummy brains. Maybe you're going to hand some of those out. But anyway, the bumps of the cerebrum, those are called gyri. One is a gyrus. And then the little grooves in the brain that we see are called sulci. One is a sulcus. And then there are deeper grooves that are called fissures. So when we're looking at the lobes, we may see a couple of fissures that are really deep. Sulci, basically. They're really deep grooves. All right, so bumps and grooves, just like bones, right? Bones have a lot of landmarks that are bumps and grooves. So anyway, a couple of more items with that we'll be seeing in these areas: the cerebral cortex, or cortices, is the plural, and then we'll look at the primary motor and primary sensory cortices as well, and we'll also be able to distinguish those with some landmarks. There'll be some sulci, fissure, gyrus landmarks that'll help us to distinguish the primary motor and primary sensory cortices. And then we'll also be using some of these as landmarks as well, the frontal, parietal, temporal lobes. And then we'll see the little brain in the back called the cerebellum. And the cerebellum is toward the base of the skull. It's going to be separate from the cerebrum. So this is a separate structure or separate feature. The cerebellum, when we dissect that, we're going to see that the inside, it's kind of a grayish color on the outside and a white color on the inside. And you're going to see what looks like kind of a tree or a bush kind of appearance in there. And they call that the arbor vitae, which is the tree of life. Some of you might have arbor vitae bushes in your yard. They're those kind of tall, a lot of folks use them for uh, almost like fencing, or privacy. The arborvitaes are really, really pretty. You can buy, they're prolific too. You can buy one uh, and a 2 footer, and in 5 years it'll be 10 feet easily. They grow really well, and they're great for um, privacy. Let's see. So anyway, moving on. We're going to dissect the brain. So most of this stuff here, we're going to be able to see without doing a heck of a lot of anything except for the removing the dura mater. Once we do that, all of these items we're going to be able to locate and identify. The rest of these will definitely want to do basically it's a mid-sagittal slice. So we're going to divide the brain or separate the brain into left and right halves, and that's going to include the cerebellum as well as the spinal cord. So your first major cut is really going to be slicing the brain right down the longitudinal fissure or right down the middle. It's separated into left and right halves. And when we do that, we're going to see the pineal gland. Before we do that, actually, we'll see the spinal cord. Some of the brains have a very tiny spinal cord coming off of them. Well, all brains, when they're in the sheep, are going to be similar in size, but the brains we're going to be working on, depending on how they were removed, you might have a 2 inch bit of spinal cord. You might have a 1 centimeter bit of spinal cord. So anyway, you should have some though. And we'll see the white matter surrounding gray with the spinal cord, which is the opposite of what we see with the rest of the brain. We'll also notice the brain stem and the pons, medulla, and the midbrain. So basically, going up from the spinal cord, our first stop will be the medulla. And then we'll see the pons, which is kind of a P-shaped, a little pouch. Pons, I think, means pouch. So you'll notice a little pouchy outcropping of the brain stem. And then, of course, then the midbrain is going to be the last section. And now we're going to be kind of inside, a little bit deeper into the brain. Actually, even before we cut the brain in half, we'll definitely be able to see the spinal cord. We may be able to see a little pineal, but we'll definitely be able to see the medulla and the pons. But then the midbrain, hypothalamus, and thalamus. The thalamus will definitely have to cut the brain open to see that. But the hypothalamus, which is inferior below the thalamus, this is where the pituitary. Where's that? Right there. So the pituitary is connected with the hypothalamus. So if you recall in your study of the brain, you may remember that the hypothalamus and pituitary are intrinsically connected, both physically and chemically. Um, when you study endocrine, if you haven't done that already, of course, you'll run into those. So there are three endocrine uh, two glands in one region, the pineal gland, the pituitary gland, and the hypothalamus are all aspects not only of the nervous system, but of the endocrine. All right. So, anyhow, we'll see these before we cut and certainly after we cut. Once we cut, we'll definitely be able to see all of these features: corpus callosum, choroid plexus, the third, fourth, and lateral ventricles, Septum, pellucidum. That's septum, of course, means wall or divider. You probably know that, like the nasal septum. Um, we see the interventricular septum when we look at the heart, but it's basically a wall. And then pellucidum, we see this in the middle. Lucid it basically means a clear window. So this is kind of like a window, basically. It's a clear wall. So you'll be able to see the septum pellucidum as well. And then lastly, the fornix. So anyhow, those are the structures that you're going to see. I'm not going to necessarily go through this slideshow mainly. Well, I don't know. I don't want to spoil the fun for next week. You can certainly go through this yourselves, but there are some items that we're gonna. This is what we're gonna be doing next week. So during the dissection, I'll have this PowerPoint pulled up, and then we're gonna basically be going right down the line through each of these slides. So between now and then, a week from now, if you would like, you can certainly go through and look at this PowerPoint on your own. But like I say, um, I don't want to be too redundant, but I also I don't want to spend any time in class next week going through this list, I guess is what I'm trying to say. So, whereas I do want to spend time in class next week going through the PowerPoint. So, or the Google slide. All right. So that's that with the sheep brain. Now let's move on where you have the same thing with the cow, or the cow eye. Not the we're not dissecting the whole cow. Um, but anyway, cow eye dissection checklist, and then the cow eye dissection PowerPoint. So let's go to the dissection checklist first. I'm going to go ahead and X these out. There we go. Well, I'll just leave this in here. I don't necessarily need to open it up in a different window. There we go. Okay. You've maybe dissected a cow eyeball before. There are just a handful of features that we're going to look at. Oh. Uh, First of all, when you get your eyeball, it's going to be covered in muscle. We call these with the eyeball, they're called extrinsic muscles. So exo means outside of. So the outside of the eyeball is covered in what we say are extrinsic muscles. Now, what they're responsible for is movement of the eyeball. So you know that your eyeball, maybe you don't, maybe nowadays we're all kind of locked in with screen. I know I am. I mean, I'm always on a screen, whether it's my computer or the TV or my tablet or my phone or I'm driving. You know, I mean, it's like constantly looking forward. So, and with glasses on, you move your eyeballs side to side, up, down, and around, I can't see as well. So anyway, we're kind of trained anyway to look forward. So, but the eye muscles, those extrinsic eye muscles, are really there to be able to move the eyeball in any direction. So we'll see there are many um, fiber directions of these different muscles. So it's kind of cool because you don't really we don't dissect any animals. We'll do a little bit with the GI, or I mean the reproductive of the rats. But when it comes to skeletal musculature, we don't really dissect anything that has that. So the eye, except for the eyeballs. So we will get to see a little bit of fascia. We'll get to see some separation of the fibers. So we'll pay attention to fiber direction. We'll even kind of separate maybe a little bit some of those muscles as we're removing them. So you can see, again, kind of the intricacies of how skeletal muscles work. We don't necessarily consider those skeletal muscles per se, even though they are. They do attach to the orbit of the eye, and they certainly have names, just like our skeletal muscles do. So, but I guess what I'm saying is we don't think of them as like moving a limb or an appendage. But yep. So you'll see some skeletal muscles, and let's see. So the first thing we have to do is remove all of that skeletal muscle. So we want to just be focused on the eyeball itself. The eyeball will also have, you remember when we looked at the brain, I mentioned that optic chiasm. We also see, we'll, we'll see the end. So that's the beginning, I guess, if you will, of the optical nerve aspects that travel to the brain. And that's for the sheep brain. But then when we look at the cow eyeball, we'll get to see the end of the optic nerve where it actually plugs into the eyeball. So when you're looking at the musculature of your eyeball when you get it, and you're kind of peeling it back and trimming it up, and oh, you also there'll be some fat, there'll be some whitish yellow is always fat, and then kind of a reddish gray, brownish is going to be muscle. So anyway, whitish, yellowish is always fat. Reds, browns, grays, typically musculature or glandular. So, so anyway, we're going to look for that. But you'll also notice, remember that nerve is covered in fat. So nerves oftentimes are a little bit different color than everything else. They're kind of a yellowish, whitish, cream color. So you're going to notice as you're getting rid of that musculature around the eyeball that there's kind of a cream, whitish, kind of a tube, like a solid tube, kind of coming out of the back of the eyeball. So when you get your eyeball, you'll see the cornea will be present, and you'll also notice the sclera. So surrounding all of this, so the muscles surrounding the majority of the eye, but you will notice a distinct window that is the cornea. And then you'll also, as you're getting rid of the musculature, you'll also notice kind of the whites of the eyeball. That's going to be the sclera. And then in the posterior aspect, you'll have that kind of, kind of tube like item kind of coming out the back. It's going to be like a cream shape. That's the optic nerve. So you'll make note of that. You don't necessarily want to trim that off as you're getting rid of all the muscle. In fact, I would say leave the optic nerve alone. And then once we get all that musculature, we identify these two features, the sclera and cornea. We're going to get to the aqueous humor, which is actually inside of this little chamber. So the eyeball is a double chambered organ where you have an anterior chamber and a posterior chamber, or a front room and a back room. The front room is smaller; it's more like a little foyer, and that's going to be between the cornea and, in essence, kind of this group here. The iris, pupil, and lens are kind of the wall that separates the front room from the back room. So that's what we'll notice. Aqueous means, of course, watery, right? Like aqua in Spanish, so or Latin. So aqueous means watery. Humor has to mean doesn't mean funny. It should, but it can and it does, but not in this in science. Humor has to do with a medium of some sort. So this is a liquidy, watery medium in essence, is what this means. So after you get all of the muscle off, you're going to make an incision through the cornea. And you'll notice too with the cow eyeball, the cornea is extremely thick. And it's because they spend a lot of time outside. Their eyeballs are pretty big. You get a lot of dust and other things. So they want to protect the rest of the eye. So the cornea is pretty thick. Relative to our eyeballs, they're extremely thick. Now, once we cut through the cornea, or we're going to make an incision through the cornea to empty out the aqueous humor. Okay, and then after we do all of that, we're going to come around and we're going to make an incision and we're going to cut the eyeball like basically kind of a in not in half per se, but I think more or less maybe like 65-35 or a third. So we're gonna because remember the front room is smaller than the back room, so we want to separate the two rooms. So we're gonna go about a third of the way back from the cornea, and we're going to make an incision with we're going to cut in with the scalpel, and then we're going to take our blunt scissors and we're going to cut around. And so we're going to separate the front room and the back room. And when we do that, we are going to find the lens and the pupil and the iris. The iris is actually the color of the eye. So the cows have kind of brownish, darkish colored eyes. So their irises are going to be a dark color. And then the pupil is actually an empty space. So the pupil is a space within the iris. So the hole in the iris is the pupil. And then the lens is going to be a pretty thick, white, clear, yellowy orange. The all of your lenses will be a little bit different, but for the most part, they're kind of cloudy, and they're like a giant skittle, um, uh, like a giant clear, kind of a yellowish clear, cloudy skittle. That's what the lenses look like. So we'll pop those babies out. And then we get to kind of that posterior aspect of the eye, and we have another humor, so another medium. Vitreous kind of has to do with gelatinous. So this is kind of a thicker gelatinous medium. So it'll be very evident when you see it. Um, it's kind of like clear slime. And then, so we'll dump that out. So we'll separate the iris pupil lens, and then the vitreous hue. This makes up kind of the wall that distinguishes anterior and posterior chambers. So now we're into the posterior chamber, which is quite large, and again, makes up probably at least two-thirds of the total mass of the eyeball. So in the lining of the back of the we're going to try to keep when we cut the front away from the back, when we remove the foyer or the anterior chamber, we're going to try to keep the vitreous humor and lens kind of intact initially, so we can see how the retina is kind of fanned out and maintaining its shape. And then we're going to dump the vitreous humor out, and then you'll see how the retina just basically folds in onto itself. The retina is an extension of the optic nerve, so it's basically the frayed wire of the optic nerve. Because remember, nerves are basically wires. So when I have the eyeball, I have the optic nerve, and it's going to basically plug right into the back of the eyeball. And when it does that, and we look inside of the eyeball, we're not going to see the nerve like this. We're going to see the nerve frayed. It's going to be just kind of opened out like an umbrella. And that vitreous humor keeps that umbrella basically open, keeps the retina frayed. And the retina is the extension of the optic nerve, so that's where all of the receptors are for light. That reaches that back of the eyeball. So the retina receives all of the information. We'll see the optic disc associated with that as well. Um, the entry point for all of this information that's going to reach the brain eventually. And then we'll also notice when we remove the vitreous humor and the retina, we'll notice the optic disc as well as this what's called tapetum, and that's going to be unique. It's like a fingerprint from the animal. So every single cow eyeball is going to have a little bit different look to the tapetum, which is kind of it's really pretty. It's the kind of the inside, it's like looks almost like the inside of a shell. It's like there'll be some aqua, blue, green shades in there, a little bit of maybe even a little bit of purplish, bluish, violet, So yeah, that'll some of them are just kind of dark, kind of a dark violet color. So we'll look at all of those. It's kind of it's kind of the pearl in the oyster, I guess, is seeing what the tapetum looks like. All right, and then these are other items I've mentioned before. I guess these should probably go on top because this is really kind of the first things we do, right? The extrinsic muscles, looking at the fat, and then also noticing that optic nerve sticking out of the back. So anyhow, that is the eyeball. So the brain and eyeball, we're talking, I don't know, let's see, it's like four, three, talking like 10, 12 structures with the eyeball, and then only maybe another, um, 15 or so with the brain. So the brain and eyeball will take us probably around an hour at the most to complete. And then we'll move on to possibly blood typing. We'll see. I may save if Andrew does have some kits, I'll save these till the end. We'll probably keep our dissection groove going. Um, and then um, and then at the end, because then we have to put the equipment away and we have to get it back out. So if we do have blood typing kits, then we'll do this at the end. All right, so let's move into the pig heart. And again, there's a PowerPoint that I'll have up, but we're going to look at the checklist. There are, I think about 15 to 20 items here with that. Pop that up a little. I'll go 200 even. There we go. All right, so when we look at the heart, um, our hearts are going to be covered and our cow hearts as well. These might be pig hearts. Andrew, he gets both. So I have pig heart. They could be cow heart. They're not much different in size. They're actually very similar in size to our hearts. They might even be slightly larger, especially if it's the cow hearts. But anyway, cow hearts, pig hearts, human hearts are fairly similar in size and appearance. Actually, there are subtle differences because, of course, they walk on all fours, and so the positioning of some of the arteries and veins is slightly different than ours, but not a lot. It's pretty uncanny. So anyway, that's good for us because then we can really get a good look at the kind of what's going on and have a nice size specimen. So the hearts are covered in pericardium. And by the way, after we do heart, I don't have them in here. I don't have a dissection checklist or uh, PowerPoint or Google Slides for the sheep pluck, the sheep digestive tract, or the kidneys, primarily because the pluck and the digestive tract, those are demos in essence. So, and they're just a couple of structures. So I basically just demo the pluck, which is going to be trachea into the bronchi, into the lungs. And then the heart, the reason I'm talking about this is because it involves the heart. The heart will also be there, the sheep heart. It'll be, right in between the lungs. And then you'll see the pericardium is usually cut and kind of the heart's kind of hanging out, but there'll still be some pericardium there. But then we'll also see, which is cool about the pluck, is the diaphragm. You can see I'm kind of leaning over, but right here you see the lungs and the heart, and then in between the lungs and then the liver and stomach is going to be that muscular diaphragm. So we'll also see when we have the pluck the thick diaphragm. It's going to be just inferior to the heart and lungs. In fact, you'll notice the pericardium is actually connected with the diaphragm. So when the diaphragm and heart and the lungs are as well, the lungs are attached to the diaphragm too. So when the diaphragm flattens out, it actually kind of pulls the heart and pulls the lungs with it. And then usually the liver and possibly a bit of the stomach, but definitely the liver it tends to be included with that pluck. So I don't have a checklist, but it's just a couple of things that we look at, and I will demo that for you. You each don't have your own pluck. Um, and then what else? Oh, the GI tract. I'll pull that up in a second. There's just a couple of features for that as well. Same with the kidneys. There's just a couple of items that we look at, like literally like three or four. All right. So anyway, so when we look at the heart, the pericardium will be there. So we're going to remove that. And once we remove the pericardium, we'll notice the base and apex of the heart. Apex, of course, means point. So in this situation, the apex is going to be toward the bottom. So if we have, oh, it disappeared. That was a cool magic trick. Can I do it again? Yeah, I don't know. Oh, it's probably some Illuminati weirdness. So I don't know. Probably put this on YouTube and get banned. But the heart's kind of shaped like this. You can see in the, I'll just point up here. There you go. You see the point of it's right here. This is the apex. And then the base of the heart is actually going to be superior. So it's like an upside down pyramid, basically. So we'll make note of that. And because the apex in that area, that's going to be the ventricular portion. That's going to be the thick, meaty portion. That's the main pump. Whereas the base up toward the top, the superior aspect of the heart, that's going to be where all of the major vessels come out, as well as where we'd find the atria. So anyway, it's important to get kind of your bearings when you have the organ in front of you. Am I looking at the front? Am I looking at the back? Am I holding it upside down, right side up? Left wise, right wise. How the heck am I holding this thing? What am I looking at here? So that's the first thing we really do is get oriented with kind of the directions in the heart. So we're going to be looking at it with the apex kind of facing us, and then the base will be away from us. So that's how we'll have it. And then the other thing we'll notice separating front from back of the heart, the front or the anterior aspect is going to be pretty much clear of everything. It's going to be real meaty. Here's a heart right here. We're looking at the front of the heart. And you can see it's basically just a lot of ventricle. Now, if I were to turn that around and we were to look at the posterior aspect or the back of the heart, you would see that there's usually some fat back there. There are several blood vessel openings and blood vessels themselves. There's just a lot more chaos, I guess, going on, a lot of gunk and a lot to try to make sense of. If you're looking at the heart and you see all of that, you're looking at the back of the heart. When you're looking at the front of the heart, it's just not a lot going on. So so those are a couple ways. So we want to look at the front of the heart or the anterior aspect initially, which again is just gonna, and then we want the point or the apex facing us. So we'll keep that. And of course, I've got the slideshow. You'll be looking. I know it may seem kind of putsy at the moment, but it won't be next week. We'll also use the aorta as a major landmark. That's the largest blood vessel in the body, and that's going to be at the base of the heart or the top of the heart. So we'll see that as well. Also, toward the base, we're going to notice the right and left atria. Or auricles are going to be like auricle means ear. So we'll notice the atria are going to have kind of little ears coming off of them. The atria, I think what surprises students most about the atria is that they're quite a bit smaller than maybe than you would suspect or you would expect. So anyway, and the other thing about the atria is that they're kind of pale in color. For the most part, the heart is really pretty red. It's pretty brick red. Um, but the atria, they tend to be kind of a whitish, grayish, light, maybe a little bit of streaky redness to them, but they're a little more whitish, grayish color. So the atria, and again, they're going to be up closer to the base. And then the majority of the heart, oh, and how do I know if I'm right or left? Again, we're going to be looking at it initially, so the right side of the heart will be on your left side. Okay, so when we're looking at the heart, we're going to be looking at the anterior aspect with the point facing or the apex towards us. So the right side of the heart is actually going to be on our left side, and the left side of the heart is going to be on our right side. So when we're looking at that, we'll pay attention to that next week as well. The two atria, again, there's not a lot to them. And then the two ventricles, however, there are a lot to those. And the right ventricle, which again, from your vantage point when you're looking at the heart, will be on your left side. And then the left is we're going to notice is a little smaller when we cut open the heart than the left ventricle. The left ventricle is very large. It's the biggest chamber of the four chambers, and it's going to have a very thick wall that's going to eject blood out to the body. So the right ventricle, basically, when we look at blood, the pathway of blood flow through the heart, that's really the important aspect of understanding the basic anatomy of the heart. So the reason we go through these, and I'll be talking about this next week as well as we're going through each of these anatomical features, is that the path of blood through the heart. So the right side, and we do that by separating right half from left half. So the right side or the right half of the heart is actually receiving blood that's low in oxygen, and it's receiving that blood from the body. And all of that blood is going to go into the right atrium first. So the right atrium is receiving blood from the body, and that blood is going to be low in oxygen. Um, the other thing that we'll see then is the right atrium dumping blood into the right ventricle, and then the right ventricle is actually going to eject blood out to the lungs. And then that blood is going to get oxygenated and then it's going to get sent back to the heart. But this time it's going to go to the left side of the heart. So the left side of the heart receives blood that's high in oxygen and it receives that blood from the lungs. So the right side receives low oxygen blood from the body. The left side of the heart receives high oxygen blood from the lungs. And then the right ventricle, or pardon me, the left atrium receives that blood from the lungs, and then it's going to dump that blood that's high in oxygen down into the left ventricle. And then the left ventricle will eject all of that blood out to the body. So the highly oxygenated blood gets ejected from the left ventricle and travels out to the body. So those are the basics, just those four items. But we're going to see the aorta is the main artery that comes off of the left ventricle. So we'll see it receives a lot of pressure, a lot of blood pressure against the aorta every time the ventricles contract. So when that left ventricle ejects blood out to the body, that blood is going into the aorta. So we'll notice how thick the walls are to the aorta. So part of receiving all of that blood and all of that pressure from the left ventricle means we have to have a thick wall to accept all of that pressure. All right, and then we'll see. So then we'll see all of this before we do any incisions. So once we get rid of the pericardium and we get acclimated and oriented to our directions and our basic features on the external aspects, at that point we can start making some incisions. So we're going to try to separate the heart into front and back. So anterior, posterior. So we're actually going to start cutting from the apex and again try to get the heart so we have the front of the heart and the back of the heart separated from one another. And at that point, we'll be able to see the insides of all four of these chambers, as well as the valves that separate or distinguish the chambers. So next up, notice we have four chambers. We're also going to then have four valves. The path of blood goes from chamber through a valve into a chamber, and then through another valve and out of the heart, and then back to a chamber, through a valve, into a chamber, out through the heart, through another valve. So chamber valve, chamber valve, chamber valve, chamber valve. So that's the path of flow that we'll see next week is going to involve that kind of movement. Chamber valve, chamber valve. There we go. So the first two valves we see here are the trike. So again, four chambers, four valves. The so as we follow the path of blood flow, we get into the right atrium first, and then we're going to go to the right ventricle. But we have to pass through a valve because it goes chamber, valve, chamber. So the first valve we see is the tricuspid. And again, when we see tricuspid, we want to remember this is the right AV valve. A for atrium, V for ventricle. So the right side AV valve, or the valve between the atrium and the ventricle, we call that the tricuspid valve. Just it's got the TRI. You can't spell the word right without RI and T. So that's how I always remembered tricuspid is on the right side. Okay. And again, it's an AV valve, atrioventricular valve. All right, and then I'm going to jump down here to number 11. The as we leave, so anyway, we go from right atrium, tricuspid valve. So right atrium's the chamber, tricuspid it's the valve, into the right ventricle chamber, and then we leave the heart, the right side of the heart, we have to go through another valve. And remember, the right side of the heart is pumping blood out to the lungs. So we're going to see then another word for lungs is pulmonary. So the exit valve from the right ventricle is going to be called the pulmonary valve. So again, we go right atrium, tricuspid valve, right ventricle, pulmonary valve. And then we're out of the heart and we're heading out to the lungs. And then we come back to the heart into chamber valve again. This time it's left atrium, still the atrium, but this time the left side because we don't want to mix low oxygen blood with high oxygen blood. Remember, the right side has that low oxygen blood. We send it out to the pulmonary, out to the lungs. We exchange carbon dioxide with oxygen. Now my blood has oxygen in it. I need to send it to the other side, the left side. So then we go into the first chamber, left atrium, and then we have our valve, our AV valve, atrioventricular valves. The left AV valve is called the bicuspid valve. They also call this the mitral valve. I should put that over here, mitral. You may hear that as well. Mitral and bicuspid are the same valve. Um, so anyway, I go from the left atrium through the bicuspid valve. Actually, the bicuspid valve is a trapdoor that opens up, blood dumps down into the left ventricle. And then again, I have to go through another chamber valve, and that valve is called the aortic valve. And again, it's because then blood is going into the aorta. So that's the next stop there is after we leave the left side of the heart, we go into the aorta through that aortic valve. So again, chamber valve on the right side, chamber valve on the left side. Okay. A couple things we'll see inside of the heart. We'll see, oh, by the way, a valve anywhere in the body, heck, anywhere outside the body, plumbing and whatnot, valves prevent backflow. So the structure is it's a valve. The function is it prevents backflow. Okay, so we don't want blood going backwards. We want to keep the line moving. So that's the purpose of the valve. So when the right and left ventricles contract, they're ejecting blood through, respectively, the pulmonary valve on the right side and the aortic valve on the left side. We don't want that blood going back up into the right atrium and left atrium. We want it to go forward, not backwards. So the tricuspid and bicuspid valves are closed when we eject blood from the ventricles. So valves prevent backflow. We'll see that the valves are connected with some special uh, cords. So we'll see cord A, tendon A in there as well. Let's see. Oh, we'll also notice that there's a wall or septum, right? We already saw this term earlier today. Septum. Right? Septum means wall. So there is in fact, you'll see the term interventricular oftentimes. So interventricular septum. So between the ventricles is a wall. Very thick wall. You also notice too the thickness of the left ventricle versus the thickness of the right ventricle when we dissect it, kind of going backwards. And then lastly, we've got a couple of blood vessels. So everything up to this point has been heart features and heart structures, except I guess the aorta. The aorta was our first vessel, but we also have some other vessels here. By the way, when we study vasculature, the cardiovascular system, cardio means heart, vascular means vessels. We have three types of vessels. We have arteries, right? Capillaries and veins. You've probably heard of all of those, or arteries, veins, capillaries, however you want to remember them. But just know you have three different blood vessels, and they're arteries, capillaries, and veins. Now, capillaries are microscopic, so we cannot see them without a microscope. So, veins and arteries are macro or gross, so we can see those with the naked eye. One thing to remember about arteries and veins and blood flow: arteries always go away from the heart. A. Artery always away. No questions about it. Okay? That means veins always towards. So and again, if you remember one, the other one's the opposite. If you remember for the rest of your life, arteries always take blood away from the heart, then you'll always remember that veins always bring blood to the heart. Okay? Okay? Capillaries are the junction between the two. So arteries are delivering blood away from the heart or taking blood away from the heart. The point is, we need to exchange nutrients. We exchange gases to tissues when we're leaving the left side of the heart. So when we take oxygenated blood away from the left side of the heart, we're taking that oxygenated blood as well as the carbs, proteins, and fats, and vitamins and minerals, all the nutrients are going to leave the left side of the heart and they're going to travel out to the body. And then all the cells of the body are going to receive those nutrients. They receive those nutrients via capillary networks. So arteries are completely enclosed. They're a closed circuit. This whole thing is a closed circuit. Now, when we do get to tissues, we see that capillaries, uh, the plasma of the blood, it contains different particles that we need. Again, carbs, proteins, fats, vitamins, minerals. And then the red blood cells with the oxygen. All that stuff needs to get dropped off to the cells. So the capillaries are where that happens. Okay. And then when we leave the capillary network, we're actually taking waste away from the cells and the tissues. So, and we're putting that waste into the venous blood supply. So veins always go toward the heart, but veins generally are going to be carrying blood that's low in oxygen, high in carbon dioxide, whereas arteries generally are going to be carrying blood that's high in oxygen, low in carbon dioxide. So remember, artery is always away, and if it's a left side of the heart artery leaving, then it's taking oxygenated blood to the body. And then if it's a vein coming back to the right side of the heart, it's bringing blood that's high in carbon dioxide. Okay. So we do have two exceptions to the veins carrying blood high in carbon dioxide. Remember, veins always go toward. So the pulmonary veins are going towards the heart, but they're going toward the left side of the heart and they're carrying blood that's high in oxygen. So the pulmonary veins are the only veins in the body that are carrying blood that's high in oxygen. All other veins are carrying blood low in oxygen, except the pulmonary veins. Because remember, the left side of the heart is receiving blood from the lungs. That blood is full of oxygen. And remember, vessels that are going toward the heart are always veins. The only difference between this vein and the rest of the veins in the body is that the pulmonary vein is carrying blood high in oxygen, okay? You have to remember that, which also means that the pulmonary arteries always away. So the right side of the heart, remember, it has all that low oxygen blood. The right ventricle, when it pumps blood through that pulmonary valve and into the pulmonary artery, that blood is going away from the heart and it's low in oxygen. So it's pulmonary artery is the only artery in the body that has low oxygen blood. So when you're asked which of the following arteries is different or which of the following veins is different, it's always pulmonary. All right. Pulmonary artery, only artery carrying blood low in oxygen. Pulmonary vein, only vein that's carrying blood that's high in oxygen. So generally, remember, veins are bringing low oxygen blood to the right side of the heart. Arteries are generally taking high oxygen blood away from the heart. All right, so we'll see some pulmonary veins and pulmonary arteries. Um, we also will notice these two here, the two vena cavae. These are the two veins or vessels that carry blood to the right atrium. So when we're looking at the right atrium, we're going to notice these two vena cavae. So the superior vena cava brings blood that's low in oxygen from above the heart to the right atrium. So the inferior vena cava is bringing blood that's low in oxygen from lower than the heart or inferior to the heart. So most of the body is sending blood low in oxygen back to the right atrium via the inferior vena cava. All right, and then arteries always away. So we have coronary arteries. These are a little bit different. These are the main ones that wrap around the heart muscle. So we're going to see these branch right off of the aorta. So when we leave the left ventricle and dump blood, go through the aortic valve and into the aorta, the first spot we see, or the first branch we see, is the coronary arteries. So, and we'll see those wrap around the heart muscle. The heart feeds itself first, so which is smart. If the pump is going to pump, the pump needs oxygen. The muscle cells of the pump need oxygen and glucose. So the coronary arteries deliver oxygen and glucose to the muscles of the heart. So these guys feed the heart. That's why if you have high cholesterol and a lot of plaque in your coronary arteries, you're decreasing the diameter, and that's going to cause an increase in blood pressure. And it's also going to cause a barrier as well for getting the nutrients delivered that need to get delivered to all of the sections of the heart. So coronary arteries we'll also see. And I think that's it for the heart. Yeah. So that'll be fun. We'll dissect the heart and take a peek at all of the different valves and chambers and blood vessels. All right, pig heart dissection. All right, and then like I say, with the pluck, we'll be focused on the lungs and lung tissue as well as the trachea, basically the respiratory tree, and that includes the diaphragm. The diaphragm performs about 75% of your ability to breathe in is because of the presence of the diaphragm. Um, the sheep GI tract, all ruminant animals, so Herbivores, cows are a good example as well, and sheep. So you want to check out ruminant digestion. Um, if not, you'll see it next week. But I do have a sheep GI tract that goes basically from esophagus. Actually, it includes large intestine, colon, and even the rectum. So our focus is going to be primarily on the stomachs of the sheep. So we're going to notice an esophagus. This is the food tube. So when you swallow food or the sheep swallows food, it's going to go down into the esophagus. And then as the esophagus enters into the stomach, we're going to see all kinds of things happening here. We're going to see a giant uh, yeah, like a big bag, like a giant pouch, like a big leather bag, called the rumen. So that'll be the most prolific or prominent feature, I guess, of this uh, GI tract is the rumen. We call them ruminants for a reason, but the rumen is going to kind of hold on to some grains, the grasses, and it's going to ferment those. So food could stay in the rumen for like a week at a time. But it's just a giant fermentation chamber. Fermentation happens from the sugars that they're trying to get out of here. All right. So anyway, we'll notice the giant rumen. And then we're also going to notice right away, there's a little side. We'll see a valve here called or they call it the sphincter. The sphincter and a valve are similar. They both try to prevent backflow, but notice the lower esophageal sphincter opening up into the rumen. And then right off the side of that is going to be something called the reticulum. And the reticulum has an interesting appearance on the inside. It's got kind of a honeycomb uh, looking deal. Guards against the intrusion of large particles that slipped past the rumen that need to be broken down more. So we might, it's kind of like a mini rumen, basically. So if we get some big chunks that maybe aren't ready to leave the digestive tract, we may dump them into the reticulum to hang out for a while. Um, and then let's see, we've got the omasum. We'll notice that as well, kind of like a coin purse looking jobber. Lots of folds in there. So this is where nutrient absorption starts to take place. Um, all bodies are interested in maximizing space. So with absorption of nutrients, we want a lot of folding so we can maximize how much nutrient we absorb. So we'll see a lot of folding in there. And then we leave pretty much the main three and we head into the abomasum, which is a lot like our stomach. So we, of course, have one stomach. Ours is the esophagus into, in essence, the abomasum. So this is where your stomach acids and digestive juices start to blend and it says mingle there. But yeah, this is a hot spot that's basically the beginnings of the end for that kind of upper GI tract heading now into the small intestines where a lot of the action's going to happen. So anyway, the esophagus dumps into the stomachs for the sheep. For us, the esophagus dumps into just the stomach, and it's going to be so when we look at when we cut open each of these, we're going to see a lot of differing uh, features here within these. So they'll be very distinctive. The abomasum has rugae, and the abomasum is going to look a lot like, like I say, like our stomach. And then we're going to have several feet of small intestine. They're going to be kind of coiled up with mesentery. And then we're going to dump into the colon and then out through the rectum. Okay. So anyway, this is really fascinating. You'll be blown away at like when we look at this and any of these images, I guess these are illustrations, they don't do justice for what's inside. It's pretty mind-blowing. what is going on inside of each of these. The rumen's going to be like a bunch of bristle brushes. We're going to see coral reef, undersea adventure, really bizarre looking features inside of those. Okay, and then let's see. I wanted to, let's go in here. And then we're going to do kidneys as well. And when we do the kidneys, they're going to be injected. So they'll have some red and blue dye in them. I don't care for that. Let's see. Here we go. This is better. So when we look at the kidneys, we're going to see some distinct color differences. First of all, we're going to see the outer kind of layer here. This is called the cortex. And the cortex is this kind of darker area, and then you can see it gets lighter in between these dark kind of fanny looking deals. This is going to be what they call the medulla, the renal medulla. And then these are called renal pyramids. Each one of these kind of upside down triangular looking deals. This says renal medulla. This actually is pointing to this whole area here, renal medulla, because these are called renal pyramids. So this would be the base of the renal pyramid and the apex of the renal pyramid. So out here are the nephrons. So out here in the renal cortex are the nephrons. And so the injection of the red dye and the blue dye. Red is for arteries, blue is for veins. And remember, when blue and red come together, when arteries meet with veins, it's actually the capillary network that's between the two. So you'll see up here is the capillary bed for all of those renal arteries and renal veins kind of come, this is where everything comes together. And remember, a capillary is the only place where anything can go into or out of the bloodstream. Kidneys are giant filters, so the goal is to filter out the plasma and some of the other gunk that might be inside the plasma, as well as some gases, especially hydrogen ions. But yeah, generally speaking, that can only take place in the capillaries, and kidneys have specialized capillaries called glomerulus. And so all of those nephrons with the glomeruli are going to be out here in the renal cortex. So on our specimens, our kidneys that we're going to see that have been injected, red and blue make purple. So this outer area is going to be kind of purplish. You're going to see a lot of red and a lot of blue going in and out of this, with a lot of purple out here. And then, so the urine gets formed and dumped into these renal pyramids. And then you're going to see what are called minor calyces. Here's it says renal calyx. So this is a minor calyx and another minor calyx. These are all minor calyces. And then they're going to dump into what are called major calyces. So that's what this is, a major calyx and another major calyx. And then all that urine is going to dump into what they call the renal pelvis. Okay, so the renal pelvis, and then all the urine leaves through what they call the ureter. Okay, there's the ureter. So this is what we're going to be looking at as well. We'll finish off with actually we're not going to finish there. The kidneys only take about 5 minutes. So sheep pluck, the heart will take about 20 minutes. The sheep pluck, sheep GI, and kidneys will take maybe a half hour total. So at that point, between the brain, eye, heart, we'll be close to that 2 hour mark. And really, all I'll need to do is show you the pregnant rats and the male rats, so you can dissect the reproductive systems, and we'll pull out the rats and see how many babies are in there. And that's probably where we'll finish. So the whole thing will take 2 hours, 20 minutes. And again, if we do have blood typing kits, we can potentially do that. But like I say, well, by the time we get through everything, your brains will be pretty fried anyway. So even if Andrew does have them, I don't know if you'll even have the uh, uh, functionality to perform them anyway. But we'll see if we have them, and we'll do them. But if not, McGraw-Hill has them, so you'll do them and understand them that way. To if you haven't done those already. Oh, by the way, here's the nephron. This is that nephron I was talking about. So all this cortex is a millions of these nephrons. And then this is the glomerulus. This is the capillary network. That's this little ball right here. So all the red blood's coming in, delivering oxygen, delivering nutrients, and then we get some waste dumped in here and filtered out, and then the urine gets formed and dumped into these pyramids, calyces, and then the ureter. All right. Well, that's really all I wanted to do today. Is kind of prep you for next week so we don't have, 'cause, I mean, look, it took over an hour to go through all of that stuff. And that would have been, we would have been doing all that in like in class. So this way we could just get in, I'll pull up the brain slide and we're just going to get right to it. So um, other than that, you guys are doing great. Keep plugging away on your McGraw-Hill labs if you haven't been. And certainly you still have plenty of time, probably about, I don't know, we probably have about 7 weeks or so left in the semester, maybe 8, I don't know. Anyway, oh, midterm grades are due this weekend. So I'll be going through the grade book. Um, I I think probably basically if you've been doing any work, you're going to have your midterm grade will be an A. If you haven't been doing any work, your midterm grade will. So in other words, if you have less than 100 points at this up to now, then you're probably going to have like a C or a D or something like that for midterm. If you have 0 points, you'll have an F. But if you have over 100 points at this point, they've been working because everything's work at your own pace. So doing midterm grades is kind of weird. But if you've done zero, then you'll have a bad midterm grade. But if you haven't done anything yet, you're probably not watching these videos anyway. So if you are watching this, or if you're here with me now, I'm sure you're getting an A. So for your midterm grade. But anyway, keep an eye on that, and that's really all I have. I'm going to stop recording.